Good morning. In the next 15 minutes, I'm going to provide you with a corporate update on StemCells Inc. I apologize if it's a little bit of a gallop, but we'll do our best to stay on time. We are a publicly traded company. We're traded on NASDAQ, so I would draw your attention to this forward-looking statement, and I would direct you to our SEC filings. So the promise of stem cells, and particularly with regards to neural stem cells, is that these cells, which are very rare cells, have the potential to repair damaged organs in the central nervous system, and they're capable of self-renewal. So a single transplantation has the potential to meaningfully and durably restore function for the life of the patient. StemCells Inc. scientists discovered, and we are now developing, a first-in-class biologic, a human neural stem cell, which is a purified, expandable, bankable product platform. These cells do not require any ex vivo processing or manipulation prior to transplant. They are directly transplantable for allogeneic use into the central nervous system. The company has completed 4 phase 1/2 clinical trials in all 3 elements of the central nervous system: the brain, spinal cord, and the eye. There have been no cell-related safety concerns identified to date, and we have followed up patients for over 6 years and actually 5 years data post-immunosuppression. Preliminary efficacy is consistent with the published preclinical studies. So there's a considerable body of preclinical work and a very strong scientific foundation. And very importantly, we've observed the durability of these cells when transplanted into a human patient. We have evidence of migration and long-term survival, as I said, greater than 6 years and 5 years post-withdrawal of immunosuppression. We have, as I said, completed 4 phase 1/2 trials, and we are now currently engaged in 2 phase 2 trials, one in spinal cord injury and one in dry age-related macular degeneration. I'm going to focus my talk today on the first 2, on spinal cord injury and dry age-related macular degeneration. In the spinal cord, we started in the thoracic region, and now we're in cervical. And in the retinal disorder area, our focus is in geographic atrophy of age-related macular degeneration, and I'll speak some more about that shortly. Spinal cord injury, won't spend a lot of time on this particular slide, except to confirm what Jane has already said. This is a very significant unmet need, and there are 12,000 cases per year in the United States, and approximately double that in Europe. About half of these patients have complete or incomplete tetraplegia. It is a very, very costly condition for the U.S. healthcare system, and the numbers speak for themselves. What's also interesting is that a very small percentage of individuals who suffer spinal cord injury have entered employment 1 year post-injury, and there are no treatments available to restore spinal cord function. Our first study was in the thoracic region, T1 to T12. It was an open-label study, single dose, 9 months of immunosuppression. This trial was authorized by Health Canada, Swissmedic, and the FDA. Of course, in these trials, your first objective is safety, but there are a series of secondary objectives which you can see for yourself, including light touch, pinprick, temperature, and electrical stimulation. There were 12 subjects enrolled, and we enrolled patients 3 to 24 months post-injury. We enrolled patients who had complete and incomplete injury. We enrolled 7 AIS A's and 5 AIS B's. We presented the results at the joint ISCOS-Asia meeting in Montreal in May of this year, and we reported overall gains in sensory function in 3 of the 7 AIS A patients in the study and 4 of the 5 AIS B's. was remarkable was that not only were the effects noticed approximately 3 months post-transplant, but they were enduring. And we also noticed that the less severe injuries gained most pronounced results, which is kind of intuitive in a way. And electrical and heat sensation thresholds were used to confirm changes in pinprick and light touch. We were quite surprised, actually, to see that 2 of the subjects, AIS A's, converted from an AIS A complete spinal cord injury to an AIS B. So this was quite unexpected. This is the first patient enrolled in that study, a Norwegian young man who was a very avid cyclist. And you can see, again, that this involvement in this study offered considerable hope to him, and he was one of the patients who converted from a complete to an incomplete injury. The corollary of what we saw in the thoracic region could well be improved motor function in the upper extremities of patients with cervical spinal cord injury, because it's the cervical region that directly innervates motor function in the upper extremities, and that, of course, is really what everybody is after. So we initiated a phase 2 study, which we call the Pathway. It's a clinical proof of concept, randomized, controlled, single-blinded. This study has been authorized by the FDA and Health Canada. We'll enroll 52 subjects with C5 to C7 injury. We will enroll AIS A's and B's, and we will extend enrollment to 24 months for the AIS B's. We will, of course, continue to monitor safety, and we'll also be looking for efficacy endpoints. And we'll be using what's known as the INSCI upper extremity motor score, which is a well-established system for measurement of motor function. We'll also, of course, be looking for changes in lower extremities, motor score, motor level, and sensory scores. To date, we have 12 sites up and running and enrolling patients. More will follow. We completed cohort 1, which is a 6-patient open-label study involving 3 doses. We completed that in April, and we commenced dosing in enrollment in June of this year in the single-blind cohort, which involves 40 subjects. We anticipate the last patient enrolled in the study will be next year. We will be announcing 6-month interim data next month. This is the cohort 1 open label study involving 6 patients. And we'll announce the 12-month data in Q2 of next year. And then in 2017, our goal is to have the top-line results available. I want to switch now to age-related macular degeneration, which, as you know, is a very significant condition. It's the #1 cause of vision loss and legal blindness in those over age 55. Prevalence, 10 million in the United States and 25 to 30 million worldwide. Of course, we've heard a lot about wet AMD. There are several treatments that are available, but there are no known cures. And it is a very significant and lucrative market for the players in the wet AMD field. Dry AMD represents the majority of age-related macular disease patients, and there are no approved treatments. StemCells Inc. is focused on the most severe form of dry AMD, which is geographic atrophy. It represents approximately 15% of the cases, and it approximates also to the size of the wet AMD market. We presented top-line results and observations to our phase 1/2 study, which was a 15-patient study. We reported visual function in the majority of patients were stable or improved, and we saw gains in contrast sensitivity. We also had major focus on anatomic changes, where we noticed increased macular volume and foveal thickness in the study eye, which is indicative, we believe, of the health of the retina. We also assessed geographic atrophy by FAF, or fundus autofluorescence. And what was very interesting to us to note was for those subjects with a lesion size that would be eligible for our phase 2 trial that's currently underway, the majority of these patients showed a slowing of the rate of geographic atrophy, which was confirmed by 2 independent reading centers. This is the data in chart form. On the left is the study eye, and you're measuring millimeters, and you're looking at change from baseline over 12 months. And these were the phase 2 eligible patients, 5 of 15 in total in the study. And to the right is the fellow eye, and also you're measuring change. If both eyes were if the macula was degenerating at the same rate, you would essentially have all of those points on that red vertical, that red line with a 45-degree angle. As you can see, the majority of the measurements were to the right of that line, meaning that the fellow eye was degenerating more quickly than the study eye that had received the cells. One patient that you can see to the left of that line, 05021, is to the right of it in the second measurement, and they say there are both 2 sites involved here. And this was a methodology difference where one site used a different methodology to include certain aspects of the retina, and one did not. So that's a little bit of a conundrum in terms of methodology. But overall, this gave us a good sense that we were on the right track for our phase 2 study. This gentleman was one of the early enrollees in the phase 1/2 study, and he truly, along with the other patients in our phase 1/2 studies, are the real pioneers. So I'll just talk briefly about the Radiance Study. 63 subjects, 20 US sites are expected to participate. The first patients have been dosed already, and we will be looking at anatomical and also functional measurements, as you can see. I want to finish with an investment rationale and catalysts for the near term. Clearly, there are large unmet market opportunities. Proof of concept phase 2 studies are underway. And it's good to note that the phase 1/2 clinical data that we've already announced is encouraging, and it is consistent with the preclinical data that have demonstrated favorable safety and efficacy. We strengthened our balance sheet in the middle of this year, and currently I believe we have a compelling current valuation with buy ratings from 5 analysts who cover the story. Anticipated catalysts, as I said, we'll be announcing 6-month data for cohort 1 in the pathway study next month. And we will be making incremental data points available in 2016 and top-line phase 2 results in 2017. So I thank you for being here. Thank you for your attention, and thank you, ARM, for inviting us to update you on our story.